Welcome to Module 2. Humans are made of DNA. DNA are basically a script, how it shapes your looks, personality, talents, and even your quirks. These DNAs have chromosomes, 46 in total, 23 pairs. And these chromosomes have genes, and there are two types of genes, genotype, phenotype. Genotype is the recipe, that's the instruction for your DNA, and phenotype is the final taste, how your traits show up. So if these two match, then it's homozygous. If these two don't match, it's called heterozygous. And as we know, there are two types, right? Dominant and recessive gene. And now we can learn about how these genes behave. Heterogeneity, even within a diagnosis like Down syndrome, there's a massive variability in abilities, behavior, and health. Why? Because each person has a different genetic background and environmental exposure. X-linked disorders. Boys XY don't have a backup. X. If the X is faulty, symptoms express fully. If girls XX carry one faulty X but remain unaffected if the other X is healthy. And protein expression levels. A gene might be partially expressed, like playing a song at a half volume. The volume can affect whether the trait is noticeable or not. So let's go back to learning about heritability. Heritability is how much of the variability in the population can be explained by genetic difference. This was studied by studying twin study. So they picked two twins, monozygotic twins who are identical, dizygotic, which are fraternal twins. And after studying, they found out the gene influenced almost everything. Environment still matters, but not equally. Genes and environment interact, and most traits are polygenic. All of these are how some traits of how genes behave. And this can be studied with epigenetic. It's a study of how environment experiences can turn gene on and off without changing the DNA itself. Now let's go ahead and learn about neurons. We have around 86 billion neurons in our body, and these are basically the brain's spark plug. And the good thing about neurons is the neuroplasticity. It's the brain's superpower. So synapses get stronger when you practice. New skills, that is, dendrites grow, and new synapses are formed. So that is called neuroplasticity. And what are these dendrites and soma and axon? So dendrites are basically the inbox. They receive message, and soma and cell body process this message, and axon delivers, it's basically the unsent message. And these are what work well together, and they communicate neuron to neuron. Neuron to neuron is basically, they don't touch, they have teeny space between them, a synaptic cleft, and they send chemical messages called neurotransmitters. These neurotransmitters deliver, and they get recycled, it's called reuptake after delivering the message. And these are some of them, neurotransmitters: glutamate, GABA, dopamine, serotonin, acetylcholine, and endomorphins. So these are what are very important for our brain to communicate with each other. Now let's talk about brain. So this is a very good example of way to remember how the brain works. And so, for example, our brains have these regions, which are frontal lobe, parietal lobe, temporal lobe, occipital lobe, cerebral, and brainstem. Each have their function. Frontal lobe is basically the bossy CEO, parietal lobe is the GPS system, and temporal lobe is the storyteller. And occipital lobe, it's like a built-in camera for vision, and cerebellum is the dancer, balance, and coordination. Brainstem is basically life function, the life support. Welcome to Module 3. We will learn about sensation perception. So sensation is your body detects stimuli like light, sound, pressure, and different things. And there are two contrasting approaches to the brain uses make sense for sensory information. So bottom-up processing basically starts with sensory input. Top-down processing uses memories, expectations. So sensation can be measured, and this is called psychophysis, and there are different types of method that you can use. One is Weber's law. So proportional difference matters. If you add 1 gram to 10 grams, then you can notice it, but if you add 1 gram to 1 kg, probably not. Different threshold. Smallest change you can notice, can you tell if the weight is 100 grams or 105 grams? Absolute threshold. Minimum stimuli you can detect, the faintest beep you can hear. And phenomenological. Description of stimulus, that's round, cloud, and shiny thing, probably an apple. These are methods of measurements. And now we're going to talk about perception. So perception, how does this work, right? So there's a stimulus. You see something, for example, a cat. And then the second thing your brain does is you notice, so you focus on it. So sensory receptors, so basically your eyes, ears, nose, tongue, touch, and all of these are involved. And then later they send the message, which is transmission. They travel to the brain. I mean, we learned about neurons before, and that's basically our communication channel, right? So after transmission, it is coded. Signals are labeled and sorted. And then we perceive, meaning is formed, and then it changes to cognition. You recognize the cat, and remember it's going to scratch you, so now you're going to action, so you're going to try to avoid the claw. So this is all what we studied before. Now let's see what our eyes can do. So eyes have cornea, which is first bendy lens, and pupil, the whole controlling light, lens, fine-tuned focus, retina, back wall with photoreceptors, and fovea, center of vision, and optic nerve, highway to the brain, and blind spots. Sometimes you can't perceive anything there. So the thalamus relays information. Primary visual cortex gets the raw input. Then parietal lobe, which is motion space, and temporal lobe are inactive. So let's see what happens in hearing. Hearing is, you have outer ear, middle ear, inner ear, and auditory nerves. So outer ear collects sound, middle ear oscillates, amplifies basically, and inner ear cochlea turns vibration into nerve signals, and auditory nerves sends information to the brain. Welcome to Module 4. We will learn about learning. Learning, a change in behavior due to experience. It's an adaptive and helps predict and survive. Allows us to find food, avoid danger, and build social bonds. And there are two types of learning: simple learning and social learning. Simple learning can be divided into habituation and sensation. Habituation is basically decreased response to repeated stimuli. Example, you stop noticing the feel of your clothes, studied by Eric Kendall using C-SLUT. And then we have, if you want to know more about what he did to the sleeves, like it was basically, he would touch them, but when he touches them, they react very quickly. But he kept touching them for a while, and then they stopped reacting. The reaction, they got so immune to it because they knew there was no threat. But then later he used electricity on them, and which made them react even more. So even though after some time he started touching them without electrocuting them, they were still reacting the same level. And now we can learn about sensation. Increased response to repeated stimulus, especially danger and painful ones. Example, burn your hand and even warm things feel painful. Habituation is equal to less neurotransmitter released and weaker response. Sensation is more neurotransmitter release, stronger response. We learn about this in Module 2. Neurotransmitters. And now we're going to go to learn about associative learning. Associative learning, there are two types: classic conditioning, operant conditioning. Classic conditioning was the two people, Paulo and John B. Watson, who worked with classic conditioning. Paulo basically experimented with a dog, and he found out that there was accusation, learning the link between CS and US. So if you want to go in deep, I have noted down the full forms of CS and RS and neutral stimuli. So CS is basically conditioned stimulus. So then extinction. CS is no longer followed by US. US is basically unconditioned stimulus, and CR weakens. CR is conditioned response. So spontaneous recovery. CR returns after reset. And generalization, similar stimuli also triggers CR. Discrimination, ability to tell stimuli apart. So then we learn about John B. Watson and Rosalia Raya. They taught baby Albert to fear rats using loud noise, showed that fear can be conditioned, which is not ethical these days, but it was a very interesting experiment. Now we're going to go to what happens beyond learning. So let's meet B.F. Skinner. So he taught us that reinforcement increased likelihood of behavior, punishment decreased likelihood of behavior, and positive, if you add stimulus, giving candy, reward, or adding chores was punishment. And negative is remove stimulus, removing chores as reward, or taking TV as punishment. Primary, naturally rewarding, example food and want. Secondary, learned value, example money and praise. So fixed ratio, which is free coffee for every 5 purchase, was a moderate level. So variable ratio, like slot machine, had very high, and fixed interval had like weekly salary had low, and variable interval had random email replies has high. So shaping reinforces small steps towards complex behavior. Application like dog training, education, parenting, video game reward systems are some of the applications that you can use. So now we're going to learn about cognitive learning. So we're going beyond behaviorism. Behaviorists like Skinner focused only on observing behavior, but not all learning is reinforcement-based. So cognitive learning is basically one, observational. That is Bandura's doll experiment, where children watch adults act aggressively towards a doll, and they imitate the same behavior. And then we're going to learn about insight learning. So Kohler and chimpanzees. Chimpanzees figured out how to grab bananas with a tool or stacked boxes, suggesting internal problem solving without trial and error. And then we also learn about latent learning. Welcome to Module 5. We will learn about memory. Think of memory as your brain's save and retrieve system, like the cloud for your mind. So it basically does 3 types, which is encoding, storage, retrieval. So encoding is basically getting information in, like typing a note. It's effortful encoding, which is studying, rehearsing mnemonics. Automatic encoding is remembering what you ate for breakfast without trying. Keeping into safe is storage, basically like saving the note. Shallow is something that's forgettable, like appearance and sound, and deeper, which is meaningful connections, which are more memorable. And last part is retrieval, which might be episodic, getting information back out, like opening the notebook later. So it's basically recall, short answers like what's your friend's name, friend's number, and recognition, multiple choice question like face in a crowd. So memory, there are different types of memory. there are three types basically: long-term, short-term, and sensory memory. So sensory memory is like for 2 or 4 seconds, unlimited brief short sensory information like sound and light. And then short-term memory is basically like 2 to 4 seconds and 20 seconds sometimes for shorter memory, and 5 to 9 items they can hold information currently thinking about. So long-term memory is lifetime, unlimited, stores everything you've got encoded deeply. So in long term, there are two types: explicit and implicit. Explicit is basically personal events, semantic facts like I had pizza yesterday, or Paris is in France. And implicit is procedural, like skills, classification, classically conditioned responses like how to ride a bike, fear of spiders. And short term and long term have serial precision effect. First item is primary effect, last item is recent effect. So middle ones are not so lucky. You don't seem to remember exactly what it is, but the first and last seem to retain for a longer time. And if short-term and short-term memory is later encoded to long-term memory, and we learned about long-term memory. So memory can be improved using neurotransmitters. Glutamate helps strengthen synapses, and acetylcholine is the key for learning and memory, which we learned about in Module 2, and also use that information in Module 3 and 4. And now we're going to talk about these decreased neurotransmitter. What happens? So for example, in middle age or middle adulthood, some decline in episodic memory, stable semantic memory, and old age decline in working memory and recall recognition often preserved. So these are because there's a decreased neurotransmitters. And shrinking hippocampus. So what happens when we learn about brain and the structure of brain in Module 2. So hippocampus forms new explicit memories, amygdala tags emotions to memories, and cerebellum and basal ganglia is procedural memory, basically skills that we learned about. And prefrontal cortex. To Module 6, we will learn about personality. Personality, think of personality as your signature vibe, the consistent way you think, feel, and behave across time and situations. So you can describe something, describe individuals' difference, explain it, why people are the way they are, predict it, behavior across context, and improve it individually and societal well-being. So personality, how do you measure personality? So personality can be measured with self-report questionnaires, behavior measure, life outcome data, observer rating, and projective tests. So study can be divided into two, nomothetic and ideographic. Nomothetic is basically focused on general laws, traits, dimensions of people. So, for example, Big 5 research. And ideographic is focused on unique individual experiences like case studies and life stories. So I guess we can say observer rating and projective tests are something of an ideographic, and observer life outcome data, behavior measure, and self-report questionnaire can be nomothetic. So how do you study personality? Let's look more in detail with Phoeb's method of studying personality. So he says we can the few techniques, which is free association. Basically, patient says whatever comes to their mind without censorship, and the purpose of it is it reveals hidden thoughts and unresolved conflicts. And then dream analysis, interpreting dreams, the road to the unconscious, uncovers latent content, hidden meanings behind manifested content. Hypnosis, early in his career, basically inducing a trance-like state and explore repressed memories, helps patients access buried memories. Slip of tongue, the Freudian slip, errors in speech that reveal unconscious thought, believed to have leaks from the unconscious, basically. And case study, detailed ethnographic analysis of individual gave insight into unconscious conflicts, shapes, and behavior. Whereas in these days, some of these techniques are believed to be non-scientific, so maybe not accurate information can be acquired. So he also came up with structure of personality. Basically, there are three types: id, ego, and superego. Id is pleasure, which is unconscious impulsive desire, like sex and aggression. Ego is reality, rational self measures it and superego. And morality is that superego, it's internalized ideas and guilt. So some of the techniques that ego uses to reduce anxiety, often unconsciously, is repression, forgetting painful thoughts; denial, refusing to accept reality; projection is attributing your own feelings to others; and displacement, redirecting impulses. Example, yelling at your dog after a bad day. So these are some of the things that we learn about personality. So now let's dive in deeper to learn more about what makes a good personality theory. So it needs to be comprehensive, parsimonious, falsifiable, and generative. Now, personality has different levels. So the dispositional traits, or broad stable traits across time and situation. For example, the Big 5 ocean. We will learn more about it deeper later. And characteristics, adaptations, basically motivations, goal, values, copying styles, and career goals, but